Hello everybody. Welcome to the fourth Revive webinar by the Global Antibiotic Research and Development Partnership GARDP. I am Astrid Pence-Moore and I'm hosting today's webinar. Revive is part of GARDP's Antimicrobial Memory, Recovery and Exploratory Program. It is an online space to support and connect the antimicrobial R&D community. Revive aims to facilitate learning, to connect people, and to share knowledge. This webinar series is part of Revive's educational activities. In addition to our online webinars, we also organize sessions at key conferences in the field. The presentation of this webinar was part of the Antibiotic Bootcamp for Developers, Preclinical Toxicology, which we co-organized together with CARB-X at the ESCMID ASM Conference on Drug Development to meet the challenge of antimicrobial resistance 2018 in Lisbon. Today, we will first show the recording of Claire Sadler's presentation at this conference, and afterwards, she will join us for a live Q&A session. You can submit your questions at any time during the webinar by using the questions window on your webinar control panel as shown in the slide. We will do our best to respond to as many questions as possible. Should you experience any problems with the viewing of this video, it may be due to the bandwidth of your connection or a delay in your network. In that case, I would recommend to you to watch the recording on our GARDP YouTube channel. You can find the direct link in the welcome message on your webinar control panel. Make sure to join us again afterwards for the live Q&A session. Today's webinar will be moderated by Ursula Theuretzbacher from the Center for Anti-Infective Agents in Vienna. Our speaker is Claire Sadler, a senior toxicologist at ApconiX. Claire graduated from the University of Liverpool with a degree in pharmacology. She gained her master's following research into the pharmacology and toxicology profile of novel antimalarials and her PhD following research into the role of neurotransmitters in obesity. In 2007, Claire joined Syngenta as an experimental pathology team leader and health assessment indication lead, supporting the early fungicide portfolio. Later on, she returned to her original interest in pharmacology, working for AstraZeneca in project toxicology, where she ultimately led the UK discovery safety team of project toxicologists and bench scientists. For the last 3 years, Claire has been working with ApconiX as a project toxicology consultant. There she is working with numerous clients from small biotechs to large pharma, designing and implementing the preclinical toxicology programs to support their projects from early discovery to early clinical development. We will now show the recording of Claire's presentation. After this 25-minute video, Claire will join us for a live Q&A. You can submit your questions at any time during the video or afterwards.
Hello, everyone. I'd like to also say thank you very much for inviting me to come and speak at this conference. My name's Claire Sadler. I'm a project toxicologist. I have about 15 years experience in the pharmaceutical industry. I've worked across a number of different indications, in oncology, inflammation and infection. And I have some, I used to work at AstraZeneca for about 10 years, and I can see some ex-colleagues of mine in the audience. Hopefully they won't heckle me too much. I currently work at a company called ApconiX, and we are actually toxicology consultants. And just a very interesting talk from Terry, and I will touch on some of those points going forward. So in my experience working in projects for many years, sometimes the non-clinical safety is viewed as a hurdle, something that a project has to get over or negotiate towards the end of a program just to get through it. Or some may consider it just to be a box to be ticked. There are guidelines out there, there are suggested designs on studies, and you just have to fill out all those boxes, and then you're free to go. And even, in my experience, sometimes we're seen as a bit of a grim reaper. We can kill your projects dead if we have toxicity issues which can't be mitigated or negotiated past. But really what we're here to do is try and help move a project forward. We're all kind of trying to do the same thing. We're trying to get antibacterials into patients and onto the market to help combat the issues that are currently facing. And what we're trying to do really is get medicines to patients as quickly and as safely as possible. Just touching on the guidelines that Terry actually mentioned there, if we look at what they actually say we're trying to do in this non-clinical evaluation time, we're just trying to characterize the toxic effects. We're trying to identify the target organs of toxicity. We're trying to understand any dose dependence and the relationship to exposure. Is that a steep dose response curve, or do we have a bit of wiggle room there? We're trying to also understand the potential reversibility of any toxicities. And from that information, we use it to set the safe starting dose. We determine the dose range that's going to be used in clinical trials, so determine the exposure caps that you may be subjected to, and we're trying to identify the parameters for clinical monitoring. Today I'm focusing on small molecules, but I've also put the front page of the ICHS 6 guidelines, which is for biologicals. So there is different guidance for biologicals as there are for small molecules. A lot of the principles for toxicology are the same, but just to be aware that some things are a bit different. So Terry really has been talking about the regulatory side of toxicology, and from this diagram here, which is you'll be all familiar with of the drug development process, toxicology is actually one of the few functions which is working throughout that development chain. The red circle there is really highlighting the regulatory toxicology part of that chain. So looking at a compound that's been selected for development, going through GLP toxicity testing, and going through the clinical trials. And through those clinical trials of both phase 1, phase 2, and phase 3, there are different regulatory required non-clinical studies that have to be conducted to support those different phases as they progress. And although there are guidelines that give you an indication of what those studies should look like, it's really important to design those programs to meet your program's requirements. You need to make sure you consider things like the duration of dosing. Is this an acute treatment or chronic? How are you going to administer this drug? Particularly for antibacterials, the combination therapies, and when are you going to combine with other agents, and how much is known about those? Which demographic or population are you going to be going in, and when? Unfortunately, infections can hit anybody, and women of childbearing potential are an important demographic that needs to be covered. And also children, so we often have to treat infants and neonates. Again, when do you need to have the tox package to support going into that population? But today what I'm actually going to focus on is the discovery end of the pipeline. So in discovery, we have a slightly different aim maybe than from regulatory, where you've got one compound to work through and you're just trying to understand the risks. In discovery, what we're trying to do is identify any potential hazards early, try and design experimental plans to assess those, and really try to influence candidate selection so that we choose the best molecule possible to give you the best chance of success. In discovery toxicology, we work straight from the target identification all the way up to candidate selection. And as mentioned in the previous talks today, this is all an integrated process. So we have to integrate with the chemistry, the physical chemistry properties that were covered this morning, think about obviously efficacy and the spectrum and the microbiology, the DMPK properties. If you can't get it in, it's not going to work, and also the toxicology. And often if you change one element of these, it will change some of the others. So it's really about trying to find that sweet spot or the balance between all those different elements to get the best drug possible. So if we think about discovery toxicology, sometimes it's useful to break it into three areas. We have the target and toxicity liabilities associated with that. We have chemistry, and then we have patient. These are all interlinked, but you can look at them all slightly separately. So for target, aim of target assessment really is to understand the normal physiology of a target. If you're going to modulate it for a disease, what happens when you modulate it in the rest of the body? Is there some adverse event that will occur? And we can use data, in silico data, literature reviews to try and understand that normal physiology, look at things like orthologs, expression analysis, pathway mapping, the phenotypes, knockout animals and human mutations, although it doesn't always occur as they are. It gives you an indication of maybe where to look. And also anything you know about competitor compounds. Maybe somebody's looked at something before. There's also in vitro tools we can look at enzyme selectivity assays, general cytotoxicity assays are useful, and also organ-specific assays. And then as the program progresses, you may have some in vivo data. You can take information from PK data, PK studies, efficacy studies, or investigational tox studies. You just need to know what question you're trying to ask. Is there something you know about this target that makes you particularly concerned about a particular organ or a particular endpoint? And can you incorporate that into some of your early in vivo studies? Now, you may think, being antibacterial, that actually the primary target isn't so important. Often you're trying to aim to hit something which is bacterial specific or bacterial selective, and therefore a human or a mammalian target isn't really relevant. And that might be the case depending on which target you're going for, but obviously this is just a slide of normal antibiotic modes of actions or common antibiotic modes of action. And if you're going for something on the cell wall, yeah, you're probably right. You're probably not going to be so concerned about your primary target. But if you were going for something like a nucleic acid synthesis target, topoisomerases, then actually you may find that mammalian cross-reactivity is something of concern or something that you need to be aware of. And as more and more novel targets are being looked at, more consideration has to be made for this. Now, I do appreciate that, as we heard this morning actually, the movement towards more phenotypic screens is around, and therefore you're not necessarily going to know what your target is, your molecular target from the beginning. But that's something that hopefully will be built as the program progresses. This is just some examples really of, so quinolones are known to inhibit bacterial type 2 topoisomerases, but they can also have activity against mammalian topoisomerases. And the top paper there is just showing how oxafloxacin causes rat testicular toxicity. It inhibits the DNA during the meiosis in the testes. And so although it's preferentially hitting bacterial type 2 topoisomerases, it does have impact in the mammalian system. The bottom paper there is really talking about 4 quinolones, which inhibit mitochondrial DNA, because mitochondrial DNA has some similarities to bacterial DNA, and so sometimes can have a bit of an issue when you're hitting that target. In terms of a target safety assessment cascade, in my experience, it's really about trying to identify potential hazards by inhibiting any orthologs. Again, looking at that literature to try and pull that out. And then developing in vitro assays to try and understand that selectivity window that you might have. One of the difficulties in this area really is when you're looking at activity in whole cell systems, so you're looking at bacterial activity versus activity in mammalian cell, is how do you compare MICs versus IC50s? These assays, in vitro assays, are very different, their formats are different, the endpoints are different, the culture systems are different. So can you really make a good judgment of the risk from that assay? So how big a window do you need when you're looking for fold selectivity? Are you looking for 10-fold? Are you looking for 100-fold? Are you looking for 1,000-fold? And really, it's not necessarily possible to identify that from in vitro work. You really need to do further work to identify the risks, potentially doing in vivo work to understand, okay, I've got a 100-fold selectivity window. I know what target toxicity I might expect to see if I was hitting that mammalian target. What actually happens when it goes into a whole animal system? The second area to think about is your chemistry, and this is definitely a consideration for anti-infectives. So you're really trying to understand your off-target liabilities and any potential baggage that you're carrying, which may limit your ability to explore your efficacious doses in the clinic. So again, using in silico, SAR databases, historical knowledge, and of course, as the talk this morning mentioned, thinking about AI. using in vitro safety screens, and I'll talk a little bit more about potential ones to use in the next slide. And again, using in vivo studies, any in vivo studies you're doing, thinking about what liabilities you may be carrying, and is there any way you can assess them early. And here, the real aim is try and identify those potential hazards early so that you've still got time to design them out with the chemistry. So an example here is of some of the major areas for antibacterials where you may want to consider doing in vitro screening. So cardiac ion channels, a lot of historical knowledge about this chemical class and its potential to inhibit cardiac ion channels. HERG is obviously commonly known. But I'd also urge you not to just think about HERG. You need to think about the other ion channels involved in the system, things like sodium and calcium channels. You have secondary pharmacology screens. Again, just looking at general promiscuity early in a program can really help you understand the potential risks you may be carrying. And interesting, a question that was this morning about physical chemical properties in vitro screening, toxicity screening. So in my experience, if you have a compound that has high lipophilicity, then you have a very promiscuous secondary pharm profile. So that's one of the examples where the physical chem properties of the compound can really influence that off-target profile. For antibacterials, genetic toxicity is very important. We have to think about the mutagenicity, and as mentioned this morning, there's a lot of structural alerts that we understand how that links into direct damage of DNA. But also with antibacterials, there's some primary targets where they're linked with the way they interact with DNA and causing signals in the genotox assays. So again, something to understand early on. Mitochondrial toxicity, as I mentioned before, is a common toxicity with antibacterials, and again, it can be an issue. And in my experience, using a general cytotoxicity in mammalian cell, just to understand how potent your compound is versus bacterial, can be quite useful to understand the potential hazards you may encounter. And something just to note, again, top dose to be used. So most indications would use 10 micromolar in an in vitro screen as their top dose concentration. It will be well excess of where they plan to be in the clinic. In my experience in antibacterials, 10 micromolar free is not necessarily in well excess of where you may need to be for in the clinic. So when you're looking at these in vitro screens, you need to think about what that top dose concentration is, and then you need to think about the solubility of your compound at that higher doses. So again, that's where physico-chem properties can really influence how you use these assays. So again, the in vitro assays here are really screens to try and identify potential hazards early, but all of them, you can get an IC50, but it doesn't tell you the whole story. You have to follow those leads and try and understand what the risks are. And I've just got a couple of examples really of programs that I've been involved with. So as we know, MBTIs, pharmacophore is well associated with cardiovascular liabilities. And this is from the AstraZeneca group over in Boston, where looking at novel aminopyrimidines and their inhibition of topoisomerase and their liability in the cardiac channel. So on the top left, you have a compound 7, which was the HERG IC50 was 44 micromolar, which in other indications you would be happy as Larry, that's really high, but again in infections not necessarily high enough. And bear in mind for HERG as well, IC50 isn't really the relevant number, although that's the one that everyone reports. And IC10 is when you start seeing activity. They assessed this compound in vivo in a guinea pig map study to look for QT prolongation. And you could see QT prolongation was demonstrated at less than 20 micromolar free. The effective therapeutic plasma concentration that was being calculated was around 2 to 10 micromolar free. So you can see your window there between an effect and where you need to be is pretty small. And so it was considered that this would have a high risk for QT prolongation in man. So there was a lot of effort put in to try and reduce that HERG inhibition, and that was actually done by reducing the pKa, so again, how physical chemical properties link in, whilst trying to maintain the other properties of the compound. And in the bottom right corner, they had a HERG IC50 of 233 micromolar. They assessed it again in a guinea pig map assay, and at the highest dose they tested, it had no QT prolongation. This second program is from the AstraZeneca group over in Bangalore, when we were looking for an antimalarial candidate. The top left graph shows you an early compound where it was shown to have very promiscuous activity against secondary farm profile. The lines on this curve, I've just annotated some of the points. The far left point that you can see is acetylcholinesterase inhibition. And as we go through the different hits that it had activity also at muscarinic receptors and adrenoreceptors. And the solid line on that graph, I don't know if it shows very well on the screen, there's a solid line on the right-hand side, which is where they predicted they needed to be for efficacy. So basically, we were getting very high coverage of these off-targets within our efficacious range. This was borne out by going into an in vivo study and seeing if we saw any toxicity, which we did, indicating that the selectivity profile of off-targets wasn't good enough. So a lot of work was done to try and move that selectivity profile. And on the bottom graph, you can see they actually moved the acetylcholinesterase, which was the focus of the SAR, over to the right-hand side. So they took away that activity. And interestingly as well, we also increased the potency. So the solid line has moved far to the left. So as well as getting rid of one of the off-targets, they increased the potency, so they increased that window of safety. It doesn't really matter how that window is brought in, whether it's you get rid of off-targets or you increase your potency. It's just about expanding that window as good as possible. This also went into in vivo testing, where we found that we got good tolerability at doses that was required. And this third example was from the team over in AstraZeneca Bangalore as well, where here we were looking at nitroarenes and for tuberculosis. And nitroarenes are known to carry the risk of mutagenicity. Again, it comes down to their physical chem properties and their nitrogens. And they had really good promising activity, but this was obviously a concern if they were also mutagenic. So we investigated cutting down the AIMS assay, basically, to be able to look at a number of compounds early on in the program. For those of you that aren't aware, an AIMS assay, which is the regulatory required mutagenicity assay, requires up to 400 mg of compound to assess. So although it's in vitro, it's compound hungry. 400 mg of compound early in a program is a lot of compound. So we adapted the assay to be able to use only 10 mg of compound, which made it a lot more tractable, to be able to look at matched pairs and work through the SAR to reduce the mutagenicity. So coming on to the third part of when we think about the toxicology, which is the patient. So we can identify the hazards early, we can look at the target-related hazards, we can look at the chemistry-related hazards, but really we need to put those hazards into context with the patient population, and we need to think about the risk versus the benefit for that population. So in order to do that, it's really essential to understand where you're going with the program. What's the clinical plan? Are we going into healthy volunteers or are we going straight into patients? With a lot of anti-infectives, we have to go into healthy volunteers to get the PK initially. The downside of that is healthy volunteers can't tolerate a toxicity that maybe a patient who is in a life-threatening condition would tolerate. We have to think about the route of administration. Are we going oral? Are we going IV? If we're going IV and we have a toxicity which we can monitor and we take away the drug very quickly, does that also mean the toxicity disappears? If we're going orally, that might not be as easy to do. Are we treating the patients in the hospital or are we treating them in the community? If they're hospitalised, they'll be highly monitored. We can then address any issues as they arise. If they're in the community, they have a lot less monitoring and we have to think about the risks associated. We have to think about the duration of treatment. Are we treating acutely? Is it 7 days treatment for a serious hospital infection? Or is it a chronic treatment? Is it tuberculosis where we'll be treating them for 6 to 18 months? A toxicity which is maybe tolerable and have a good safety window for 7 days, because we can show things like reversibility, may be looked at very differently if it's something we're treating for 18 months. We also have to think about the age of the patient. Are they elderly? What are the comorbidities they'll also be having? Or are they young? If it's something that may affect fertility, say, an elderly patient may accept that tolerability risk, where a neonate, that may not be so acceptable. And all of this has to be put in with the calculated exposures required for efficacy. So any hazard we identify, it's really about where do we need to be for efficacy, what's the exposures required to understand that risk. And I appreciate early on in a program, it's a very difficult thing to answer. So we use surrogates, we look at MIC90s, we look at efficacy exposures, but the real assessment comes on when we can understand where we need to be in the clinic. This is a slide I quite like. It's an indication of how to look at hazard versus risk. So here we have 4 men in full protective gear looking at something in a field. I don't know what it is. They've assessed that. They've looked at that hazard, they've made a risk assessment, and they've decided they need to be fully kitted out to understand it. And then we've got somebody who's just stood there. He's exposing himself more than they, but he thinks that's okay. I don't know who's right. I don't know the circumstance, but it just shows the difference between looking at a hazard and understanding the risk of it. So one of the questions I'm often asked is, well, what do you do when? You're trying to develop a drug, you've got your efficacy to worry about, your microbiology activity, you've got your physical chem properties, you've got your DMPK properties, you've got your safety profile. It's a lot to try and shoehorn into a program where you have limited money, you have limited compound supplies, and you have limited time. And really, for me, it's about trying to understand early on, what do you think your potential hazards are? Build a bit of data, try and understand how real you think they are. And if you think they're real, you build on that some more, you add some more experiments in to understand the real risk assessment that you might have, and you try and mitigate around them. But it's an iterative process, and it's something that should be done right from the beginning all the way through to your candidate selection. Tox shouldn't be just done right at the end. So in summary, safety should be part of the whole drug design process. Identify hazards early, design experiments to understand them better, mitigate them if you can, put any risk you do find into context, and think about the risk versus benefit for your patients. And with all that, you hopefully will identify the best molecule to give you the best chance of success. Thank you very much.
I'm now happy to welcome Claire Sadler for our live Q&A session. And with this, I'm handing over to our moderator, Ursula Theuretzbacher.
Thank you, Astrid, for this introduction. I'm delighted to moderate today's discussion about one of the key areas in discovery and preclinical development. And I'm welcoming Claire Sadler to this session and discussion. Toxicological challenges are still a main reason for determining a preclinical program. It's sometimes a neglected field because there are so many challenges during this early phase of a program, especially if it comes to innovation and molecules without historical knowledge. The interest in toxicology is high, and we have already received several questions. Please feel free to submit your questions also during this following Q&A session via your control panel. Thank you to Claire Sadler for being available for these questions. And I'm starting with the first question that's dealing with renal toxicity, which is a common problem with antibiotics. So what is the current understanding, and what are the recommended available tools and assays to screen for and de-risk renal toxicity in the preclinical space?
Thank you, Ursula, and thank you for inviting me to do this Q&A session. Nephrotoxicity is, as you say, a common issue for certain classes of antibacterials, and it's a pretty tricky one, I'll be honest, to get good understanding preclinically. There have been really great developments in the in vitro field in getting better 3D and more physiologically relevant kidney models up and running that can be used for toxicity assessment. And they are, they're really useful. Some of the downsides to them is that they aren't as high throughput as maybe using things like a hex cell line, and therefore they're a bit more costly, but they do have better profiles for expressing transporters and proteins that are important in the kidney to give you maybe a good assessment. However, one of the trickiest bits about assessing for kidney toxicity is it's not just obviously about interaction with the cells within the kidney, but it's also the full ADME profile that really influences whether something is nephrotoxic or not. So understanding where your compound distributes, if it accumulates, how it's eliminated, are all really important factors when looking at nephrotoxicity. So often I think early on in a program, the best you can do is really understand how your compound, especially if it's in a class that's known to be nephrotoxic, is maybe look at your compound in comparison to other compounds and start ranking what your compounds look like to look for the more toxic and the least toxic. But then I think to really understand the risk going forward, you do need to have an in vivo assessment so you can see where your compound goes and whether it does accumulate and distribute into the kidney, because that can make a real influence in terms of how toxic something is.
So comparing a new drug with colistin maybe not the best idea if it's as bad as colistin.
Yeah, exactly.
It always depends what you're comparing it with.
Yeah.
So the next question is dealing with oral agents. Are the tox requirements the same? Are they similar, or is there anything specifically important when you compare it with systemically delivered agents?
So in the tox requirements overall are the same. What we're trying to do in the preclinical space is really understand the overall toxicity profile of the compound, identify the target organs of toxicity, understand if there's an exposure relationship to that toxicity, and then work out how it can be monitored for and whether it's reversible. So those aspects are the same. It doesn't matter the route of administration. However, there are some toxicities that might be more virulent in an oral program compared to something like an IV program, in terms of things like if you've got local irritants in the GI tract, if your compound is fairly insoluble, that you can get an effect orally that you don't see IV. But the important factor really is that when you're developing a program, and going back to, I guess, my slide in the talk about understanding your clinical plan, is that really when you start evaluating that compound, you evaluate it in the same route of administration you plan to go forward with. Again, toxicity has a very close relationship with the ADME properties, so the exposure of your compound. So if you plan to do an IV infusion or an oral administration, ultimately, if you look at a compound with say a bolus and a high Cmax, you may get a very different tox profile, which isn't really relevant for how you plan to go. So I think it's just important that in that preclinical stage, you are looking at using the route of administration in the animal studies that you do plan to go forward with into the clinic.
Another question is, are there canonical signs of mitochondrial toxicity in animals?
Sorry, are there what signs? Sorry, Ursula, could you repeat the question?
Are there canonical signs of mitochondrial toxicity in animals?
So a difficult question, actually. So if you have mitochondrial toxicity, you may well see clinical signs, but there aren't necessarily overt clinical signs that you can visualize and then say, well, that's due to mitochondrial toxicity, because often the clinical signs are too vague for that. You'll see if an animal is feeling unwell, that you'll see general signs of morbidity, so piloerection, pinched skin, subdued behavior, and all of those. None of those are directly linked to mitochondrial toxicity. There are lots of reasons why you could get those effects, so it's very difficult to say. However, there are some ones which are maybe more identifying with mitochondrial toxicity. Things like body temperature change can sometimes be more linked. Also things like it's not a clinical sign as such, but a biomarker. So things like lactate changes can also sometimes be associated with the mitochondrial toxicant mechanism. But really, I think it's if you start seeing the toxicity, you need to start doing the sort of digging down into understanding what the cause can be, because unfortunately when we start seeing clinical signs in animals, there can often be many reasons why that has occurred, and you just need to kind of do some investigative work to try and understand what the reasons are.
Thank you. Another question is dealing with high plasma protein binding. And I've seen very high plasma protein binding in many very early programs. So that clearly affects activity and also pharmacokinetics. But the question is, how does this protein binding affect in vitro toxicology testing?
That's a really good question. So it depends on, I guess, in vitro, it depends on what assay you're using and how much protein or serum is in the assay, and whether you are actually getting a skewed result based on the fact that a lot of your compound isn't available. If that the endpoint is really relying on free drug. For in vivo situations, it's again an interesting point, because some toxicities, some functional toxicities like changes in cardiovascular system, etc., are often in the community considered to be free drug related. You're hitting a receptor, so it's really the amount of free drug that's available that is interacting with that receptor, which then leads to the effect. But there are also toxicities where we don't really know if it's about free drug or whether it's actually about total drug that's just accumulating in cells. And so for those toxicities, you sometimes have to bear in mind that you have to look at what your total exposure is, not just your free. So in depending on the toxicity, some it is very much about receptor occupancy, and therefore the free levels are important. And when you look in vitro, you just need to make sure you allow for understanding that concentration range and where you're seeing the effect. But for some of the in vivo toxicities, we don't always know it's free drug that's causing the effect. It can sometimes be total.
Okay, so it's a really difficult area, also in other in vitro testing systems. Yeah, moving to the next question. It's about a hERG. So the question is the IC20 a better indicator for QT prolongation compared to the hERG IC50? You mentioned in the presentation IC10, but our attendee was not sure if 10% inhibition would be accurately measured under assay conditions. Could you please comment on that?
Yeah, so you're right. I did say IC10, and I think it's a grey area, I guess. So I definitely below an IC50 is important. So whether it's looking at the IC20 or the IC10, you are right, it does depend a little bit on the assay variability and how reliable that IC10 is. It's really about when you start correlating that effect, that hERG activity that you're seeing, if you do in vivo assessments and you're trying to understand where you start seeing QT change and how that correlates to the concentration at which you saw inhibition on the hERG channel, whether that be IC10 or IC20, to understand whether you've got a connected effect from the molecular through to a kind of phenotypic readout or functional readout, or whether it's not. So you definitely need to look at least your IC20. At the IC50, it's not necessarily going to give you the information you need, and sometimes looking a bit lower can also be important. And I think when you think about the in vivo assessment, you also have the same kind of question where some people will only take a QT change of greater than 10% as an actual change in QT, where some people will assess a QT change or prolongation of greater than 5% and say that's relevant. And again, I think you have to line it all up and look to see how your in vitro data relates to that in vivo effect you're seeing. Look at the limitations of what you've assessed in terms of both the in vitro and the in vivo assays. What's the top concentrations you've used? What's the solubility? What's the exposure? And it's really about understanding that connection between the molecular to the in vivo, because that then helps you understand how that will maybe correlate or translate to the human and the clinical situation. So definitely lower than IC50, IC10, IC20. I think is a bit more variable, but I think it's just important to understand where that is versus where you're planning to be, and to always follow up that HERG assessment with an in vivo assessment to understand if you get a significant left shift when you go in vivo or not, because that's, again, very important.
I've another related question. So could you comment on criteria for red flags in chemical classes where you would not expect this challenge?
Sorry, Esther, could you repeat the question?
Yeah. Do you know some, or could you advise on criteria for some red flags, if there are red flags raising from your program in chemical classes where you would not expect this challenge from historical knowledge?
So I guess, sorry, I'll just rephrase the question back and check I've kind of understood it right. So you're asking where, what toxicities would give you red flags and would you not want to continue a program with?
It was related to HERG channels.
Okay. So I think, so again, even with HERG inhibition and QT prolongation, again, putting it in context with where you're planning to be in the program is important. It's not, I don't think you can give an absolute number and say that you can't go forward with that. Like I say, a QT prolongation in vivo of greater than 10% is a prolongation, but the real question is, when you go into the clinic, what will you see? And even a QT prolongation in itself is not necessarily adverse. It's only if it leads to an arrhythmia that then leads to torsades du points that it's really adverse. So I think, again, it's about understanding your compound, where you plan to go. If it's say IV and you start seeing a QT prolongation in the clinic, you can stop the dosing. If the compound's cleared fairly quickly, then you'll probably be okay in terms of it's monitorable, it'll be reversible, and if you start seeing an effect, you can pull back the dosing. Again, in a patient situation, if they have a life-threatening condition and it's about them having this to save their life, they may tolerate a little bit of a QT prolongation in order to save their life. So it just all has to be put into context. I think in an early discovery program, a general rule of thumb is you want a HERG value, if possible, of greater than 10 micromolar, purely based on the fact that a lot of compounds, when they go into the clinic, don't produce a free Cmax of greater than 10 micromolar. So I think it's a general rule of thumb, but I think if your IC50 is somewhere between 8 and 15, it's really about understanding that going forward. And like I say, the other thing is, it's not about just the in vitro assay, it's about looking at the in vivo assessment and seeing if you get a significant left shift or not in terms of activity, because that obviously has a big impact too.
Yes, thanks for answering quite a complex question. Another one is, should high bilirubin levels always be a cause for concern?
Bilirubin, was that? High bilirubin?
High bilirubin levels, yes.
So it depends on what's causing the high bilirubin levels. Often high bilirubin levels are associated with liver damage, and that is a cause for concern, obviously. But then if you do start seeing high bilirubin levels, it's good to understand whether it's conjugated or unconjugated, because that could help you understand what the cause is. So I think it is a cause for concern in the sense that it needs to be investigated further. But I don't think it's necessarily something that would stop a program. It's just you really need to understand the mechanism behind it a bit better.
Yeah, we can stay within this area of liver questions. Does oral administration make liver toxicity more likely? What are the most common causes of liver toxicity in antimicrobials?
Okay, that's a big question. So oral administration, it really, again, depends on the route of elimination of a compound, whether obviously you're going to get, when it's absorbed to the GI tract, it's going to go through the liver fairly quickly, but also obviously IV, the liver is very vascularized, so it goes to the liver quite quickly there. So I wouldn't have said necessarily that an oral administration of a compound would give you more likelihood for a liver toxicity. And in terms of antimicrobial liver toxicity, there are a number of different mechanisms. You can have straight necrosis of your hepatocytes, which leads to release of the liver enzymes and leads to damage. You can also have bile transport inhibition, so then you can get build up things like the bilirubin. You can get your hypertrophy. If you've got things like CYP induction, you can start getting hypertrophy of the liver cells. So I don't recollect really that in antibiotics there is a specific mechanism related to each different mode of action. I'm just trying to think if I can think of one. But there are various modes of liver toxicity, and again, it's just understanding how your compound stacks up in the whole organism system to understand whether it's a real risk or not.
Yeah, thanks. We are moving to another very big area. Question. So how would you approach toxicity studies for relatively big peptides? And this, of course, includes immunogenicity studies. So it's a big area too.
Yeah, so I mean, yeah, when you're looking at big peptides or molecules, it's a different area, because I think sometimes, obviously, the in vitro models that we may want to use to help address some of the issues aren't necessarily going to be relevant, depending, obviously, on the modality being looked at. And when we look at things like immunogenicity against those big peptides, unfortunately, the in vivo models we have to play with in terms of rodents, etc., aren't necessarily the right models either. And so I think really in those programs, again, you need to think carefully about what you're targeting, what your mode of action is, what your patient population is, and then look at what your main risks potentially are, and then try and work through a program of how to assess that, whether that be looking at human PBMCs or something to understand your kind of some of the immunological risks potentially, or whether that be going into things like your non-human primate to try and understand a full in vivo assessment and the risks. But I think for large molecules or for different modalities, there isn't a well-trodden path at the moment in antibacterials necessarily. So I think it's about taking each individual program and trying to work through what you think the main risks are and the best ways to assess each one. And I realise that's quite a vague answer, but because I think each project is so individual, you need to look at them in specifics to understand the right route forward.
Mm-hmm. We are moving to mutagenic potential. So mutagenic potential is critical for antibacterial drugs, as you mentioned. It's important for the discussion about differences between somatic cell mutagens and bacterial cell mutagens. So how critical is it to understand the bacterial cell mutagenicity potential during drug development?
So I think even for antibacterials, it's something that is of, in terms of the regulators will ask. So I think it's worth understanding your risk there, but it's always countered with having a mammalian-based system to assess genotoxicity as well. So in terms of the S2 regulation, ICH S2 guidelines, which is the genotox specific guidelines, they recommend that for an antibacterial, although especially if you have broad spectrum activity, you may find that you kill a lot of those bacteria in a bacterial assay, and it may be a bacterial specific mutagenicity. You counter that with your mammalian cell assay and in vivo assay as well to try and understand that true risk going forward. I think it's worth understanding that you have the risk though, and working through whether it's truly a mechanism related risk or whether it is actually you've got some structural alerts in your chemistry which is making the difference, because I think that has a different view on it.
We also have a listener here who is focused on small molecule host-directed treatment. So would you have any general advice for a company doing lead optimization for such a small molecule host-directed antibacterial treatment?
No, only in the sense of, again, it's very difficult without knowing the specifics of each program. But I guess just in general terms, it's about understanding what potential risks or toxicities you think may occur by using a host-mediated mechanism. You know, are you activating an immune system? Are you inhibiting it, inhibiting checkpoints of it? What are those risks that are potentially going to occur? And then trying to design a program to understand those better, and you'll lead up through to the GLP tox studies.
Mm-hmm. Yeah, here's another question that's dealing more with interactions that, of course, can also lead to toxic problems. If a compound has a CYP3A4 inhibition liability, can it still be used in combination with an antibiotic with no CRP liability?
So it depends on whether the antibiotic it's being used with is a CYP3A4, is primarily metabolized by CYP3A4. I guess if your compound is inhibiting CYP3A4, the risk is that anything it's dosed in combination with, it's going to affect the exposure of that agent you're in combination with, if that combination agent is metabolized by that enzyme. And then you have to look at, and if it is, and you're inhibiting the metabolism of that enzyme, of that drug, and therefore you have high exposure levels of that drug, what would be the adverse consequences of that? So how close a toxicity window or therapeutic window is there in the alternative combination agent? So I think it's a stepwise understanding of what you're going to combine, what your drug does, whether it inhibits CYP3A4 and whether it inhibits anything else as well, whether it then will impact the metabolism of a combination agent of the antibacterial. And if it does inhibit that metabolism, how tight a therapeutic window does the other agent already have? So if you do increase the exposure of that, how adverse could that potentially be? And then if that is possible, then I think it's a very careful route forward in the clinic in terms of understanding from your in vitro CYP3A4 inhibition data, does that play out when you go into the clinic? So it's I think a stepwise process from an early project to understand that CYP inhibition and understand where you plan to combine. But it can be an issue when you go in the clinic if you are inhibiting, you know, major metabolizing enzymes like CYP3A4 in terms of what you can dose with going forward. So I think if you have the option to try and get away from that early on in discovery, it may be worth putting a bit of resource behind that.
Thank you. The next question would be, which QSAR tools are the best for predicting toxicological properties?
So I think depends on what endpoint you're looking at. I guess obviously there's quite a lot of QSAR models available to test things like mutagenicity. You know, there's Derek case, MCase, etc. There are databases out there where they have quite expansive chemistry sets in there that have learned about the toxicity of that of general chemistry structures, and therefore can predict what they think from your chemistry structure what will happen. I think in all other toxicities, I'm not aware of any particularly good informatic tools that have a broad enough reach to really able to understand the toxicity risks and predict what could happen from your compound based on that. I think that there are within large pharma, they maybe have their own in-house tools, which they're heavily biased towards their chemistry sets and therefore have learned what some of the risks are associated with, but I'm not aware of any really good publicly available databases for anything other than mutagenicity.
Yeah, with the next question, we are coming back to QT prolongation. The question is, what is the acceptable margin between the plasma concentrations required for pharmacodynamic effect and the concentrations which would induce clinically significant QT prolongation or similar cardiac effects?
So, interesting question. I guess, again, it depends on, and I say depends a lot, I apologize. It really depends on what your target population is. Are they going to be monitored or are they not, as to how big that window may need to be? there isn't a set guideline in terms of what the regulators will accept. It is more of an understanding of where you think you need to be when you get to that exposure limit, where you think you will be in terms of QT prolongation, and making sure you are monitoring when you're at that kind of exposure to see whether it actually comes true in the clinic or not. And again, how you're delivering the drug can affect, again, how close to that endpoint you can be, because if it's an oral drug, then you can't withdraw treatment straight as soon as you start seeing QT prolongation, where if it's an IV drug, you probably can. So there isn't a set number. I guess in terms of my experience in working in pharma and the kind of numbers that we look for, we often are looking for a 30-fold margin between the QT effect, a no effect level, so a no effect level in QT prolongation, versus where you predict your free Cmax will be in the clinic. However, again, in antibiotics, that's not always the most applicable number, because in antibiotics, we can often be at very high free Cmaxes, and therefore you might want less than 30-fold. So there isn't a hard and fast rule, the bigger the better, but again, I think you need to take into consideration what your benefit of your drug will be, who you'll be going into in terms of patient population, how monitored they will be, and how easy it will be to take them off drug or reduce drug exposure if there seems to be an issue.
Yeah, not an easy task.
No.
We are moving to a next safety concern. It's convulsion. Convulsion is known to be related to some carbapenems. I mean, we have experience with that. Are there non-invasive methods to assess pro or anticonvulsive side effects?
So in a sense there are. So in terms of, depending on what the convulsive, if it's a true seizure or not. So I guess in the preclinical world, in rodents and in animals, you can sometimes see convulsions which are not due to actual seizure, they're just due to other reasons that can lead to a convulsion. But if it's a seizure liability, obviously if we know it's to do with binding to GABA, then you can use things like a Serret panel to look for your GABA activity, and obviously try and reduce any GABA activity. The other thing to consider is trying to reduce, if it still fits with your patient TPP, but is to reduce your things like your blood-brain barrier penetration, again to reduce the risk of any effect you're having actually leading to seizure. In terms of actual in vitro models to look at, there are things like hippocampal slices you can use to look at action potential across brain tissue to try again understand your potential seizure potential. But again, these are all can be put into you have to be put into context. So any data you get from these, you need to be able to compare against either carbapenem or other agents that you know cause seizure to try and understand where you are relative to that. But then ultimately it's going in vivo and looking in vivo and seeing at what exposure you start seeing a change in things like an EEG assessment to understand whether the seizure is likely to occur in the region that you need to be in.
Thank you. Something different now. When developing a compound as a prodrug, do you need to conduct full toxicity studies for both prodrug and the active metabolite?
So no. So in vivo, if you as long as your prodrug is metabolized into the active drug within the in vivo species you're in, you can administer it as the prodrug and just as the prodrug, and then see obviously understand the toxicity profile of the compound in vivo, because you're assessing both the prodrug and the active ingredient, in fact, in that situation. In vitro, yes, you may want to do both your prodrug and your active ingredient, particularly in things like the GenTox assessments like AIMS or the MLA, because you otherwise you may be carrying a risk that you're not aware of. And the same with things like the Serret panel, etc. You may have a difference in your profiles in the prodrug versus the active drug. So in vitro, I would say you'd assess both, but in vivo, I'd say you only assess the prodrug, as long as it does go into the active ingredient in the species you're in.
Yeah, we are coming now to a an area where there are a lot of questions, and this is the topical use of antibiotics. So does local administration generally allow high drug levels to be used before systemic toxicity is seen? And related to this, what are the most commonly tested or need to be tested off-target liabilities for skin surface applied antibiotics?
So in terms of, and I don't have a great deal of experience of topical administration antibiotics. However, in terms of where you start seeing systemic toxicity, it really depends from a topical administration. It does depend on obviously the absorption of your compounds and how well absorbed and how fast it's absorbed, and how much is absorbed as to how and when you start seeing your systemic toxicity issues. But you do have to assess that. You do have to understand the again, by assessing through either by doing a study on the topical route or by doing low dose studies systemically, and then understanding how much of your topical agent actually will go systemic is important. In terms of toxicities that are particularly related, again, I apologize, I don't have a great experience in topical, so I'm not aware of any particular, apart from of course things like skin irritation, etc., but I'm not aware of any other particular issues.
Yeah, I mean, topical antibiotics is really a very special field with special requirements. Another big topic is if you are developing a combination of two antibiotics or a combination of an antibiotic and then a potentiator, or a combination of an antibiotic and a non-antibiotic. Do you need to test everything separately and also in combination, or what are the very specific requirements for developing combination therapy?
Yeah, so it depends a bit on how much you know about each of your individual agents before you look at them in combination. But if you were testing something like a beta-lactam and a beta-lactamase inhibitor, where you know your BLI is only going to be used with your beta-lactam, so you know that going forward, you still need to understand the potential risks of each agent alone before you can combine them. So you would want to understand, you would have to conduct some in vivo studies in two species of both agents as monotherapies. But when you come to the combination experiment of the agents, you don't have to do the full package again. You can look in just one species. Normally you have a fixed, you obviously need to know what your fixed ratio of your two compounds are going to be. And you can do a 3-dose level rodent combination experiment to say, well, I know what my agent A does, I know what my agent B does, and when I've put them together, I don't get anything unusual or unexpected when I've put them together. So that's why the combination part can be real cut down. It's really the agent A and agent B. As long as you understand those well, you can reduce the combination study to one species. But the difficulty is it is a fixed ratio study, so you have to have some understanding of how you are going to dose the agents together, what kind of ratio you're going to put them in, so that you get the right data coming from your safety studies.
A different question about biomarkers for toxicity in early phases. Could you please comment on that?
In just biomarkers in general, or a particular organ?
It was just in general, the question.
In general. Yep. So, I mean, toxicity biomarkers are always fairly a hot topic in terms of understanding as early as predictive biomarkers, so we can track a toxicity and understand the risk early on in studies and in the clinic. Unfortunately, a lot of biomarkers are biomarkers of damage, so they're not predictive. They are just indicating that damage has occurred. So, you know, the common, some of the common kidney markers, or markers of kidney toxicity, sorry, some of the common markers for liver toxicity like transaminases, these, or troponins for cardiovascular toxicity, these are all markers of when an organ has gone under damage and released a protein or enzyme, and we then measure that. So you have to have a certain amount of damage before you find the marker, and therefore that it's not a predictive marker. But they can be useful in understanding the time dependency of something, and they can also be useful to understand the recovery of a tissue. Again, for different organ systems, we have different markers that have been used commonly preclinically, and obviously some are commonly used clinically. It can be very useful if you have a marker that can be used both preclinically and clinically, so you can kind of track how the toxicity develops through both the in vivo stage and the clinical stage. But in my experience, there aren't an awful lot of predictive biomarkers. They normally are a marker of damage.
So a very special situation, if you have a drug candidate with a species-specific metabolism limiting exposure in rodents that is more than tenfold, how do you obtain tox data in 2 species to support the IND and phase 1 studies?
So I think there's a number of kind of questions that you have to kind of go through before you know where the situation is. So the first question is it reduced by tenfold, but is it still above where you need to be for efficacy, or is this actually reduced further so you're not covering your efficacious range in the rodent species? Second question is you need to make sure that you are covering, have you tried all rodent species? I was recently at a CRO actually where they have a lifetime bioassay in a hamster running, because the hamster is now is the only rodent species which is has got exposure for, or doesn't highly metabolize the compound of interest. So I think you have to be quite sure that when you say you can't use a rodent species, that you have really assessed a lot of rodent species and made sure that's clear. And then the question is, in your non-rodent species, what are the toxicities? Are they, is your agent a really novel agent, or are you an agent in a class where we've been before, so we maybe understand that toxicity profile well? And then it can be a conversation with the regulators in terms of, this is what you can do, this is what you've tried to do and been unable to maybe assess in a rodent species, so you've only got 1 species, and how do they view that data set? And again, I think it does come down to, well, what are the toxicities you're seeing? Is this a novel agent? Is this a known class, and therefore we understand the toxicity profile better? Because it's all about trying to understand how great a risk will this be for humans, and I think all that information can really help with that.
Thank you. Then a next probably difficult question related to interpretation of in vivo toxicity in animal models, and main no-go criteria. So that here was an example: piloerection and reduced in-cage activity of mice after IV injection, a major sign of toxicity?
No, I think they are signs that the animals aren't necessarily particularly happy, but that piloerection and subdued behavior in a cage is not enough to say that that's a kind of a red flag necessarily. I think you'd want to understand, does it get worse over repeated dosing? Is it recoverable, within a day? Do they get better again? Is it what you'd expect, knowing the mode of action of your compound, or are you in a novel area and therefore you don't know what to expect? And then piloerection and subdued behavior, you would want to know, well, what's causing it? So is that due to general morbidity and something general is going on that's causing some toxicity signs, or is it directly related to something that's happening in the CNS? And again, things like secondary farm panels can help you kind of understand, are there any key targets that are being hit that would make you question whether there's a CNS effect or not. But for me, it's not enough to be worried. I think you would want to explore that more within your compound. Is it every compound that's causing that? Is it just one compound that's causing it? So is it an inherent risk to your series, or is it just a spurious result really with one compound? And then there's the question of, okay, you've given something IV in a mouse. What volume did you give it in? What was the formulation? What was the speed at which you delivered it? Because all those things can cause some clinical signs in rodents. So I think I'd consider all those factors too.
Now a general question again, and I would be interested to hear your response. What are the most common mistakes that you see in tox programs?
Most common mistake. Oh, that's a hard one, isn't it? Common mistakes. So I think I wouldn't say I see common mistakes as such. I think the common drug development issues that you see that aren't addressed maybe as early as would be ideal are things like understanding where you're going clinically as early as you possibly can, so you can frame everything you're doing with that in mind. I think understanding things like your formulation that you're going to use both clinically and pre-clinically, again, can be an issue which if it's done too late in the day, you just can't get around. Yeah, no, I think that, and so yeah, so and again, really that that formulation is really about, again, clinical administration, how you plan to administer this. So I think for me, the commonest mistake is not thinking far enough ahead. So I think in drug discovery, you can, drug discovery and clinics seem so far away that you don't necessarily think about it. But I think, again, understanding the ADME properties you need, understanding the physical properties you need of a compound, and understanding the bacterial spectrum you need, understanding the toxicity profile that's tolerable, all of that just pins on where you go in the clinic, what your plan is in the clinic. So I think understanding that as early as possible is really key.
Thank you. Are there specific toxicity and safety studies required if you modify the route of administration, for instance, from oral to IV or the other way around?
Yeah, so if you are changing your route of administration, so you've done a tox package with one route and you're moving to a second route, you do need to look at redoing some of your tox study, not redoing, but you need to bolster your tox package with the correct route of administration. So an example would be the general tox studies. If you have done them IV initially and then you're doing an oral step down therapy, you may need to do a full package again on the oral administration in both species, because your kinetics of your drug, your AUC, your clearance, the way it's tolerated, it may change. So you do have to consider that. However, you may be able to not have to repeat things like the safety pharmacology aspects, which are more driven on a Cmax basis. As for an oral administration, you probably will have a lower Cmax than you did for IV. So again, it's about looking at your program, what the main key areas of concern, what the toxicities you saw. Do we understand the PK driver? Is it Cmax or is it AUC? And therefore, what package do you need to put together to try and get into the different route in the clinic? But I would say that, again, in my experience, regulators, particularly in this area, are very keen to talk. They don't want unnecessary work to be done. So if you're not sure and you need that scientific advice, I would go to the regulator you plan to go to and ask them for their advice as to, with the package you have in hand, what else would they like to see when you change your route?
Thank you. So now I'm coming to the last question that I can ask you before we close this session. It's more a philosophical question, I think. And of course, there is no clear answer to it. But one of our listeners would like to know, what's your opinion? What is causing the difficulty to identify novel antibiotics against gram-negative bacteria? So is it more due to lack of pharmacological activity, or is it more because of these high toxicological challenges? Yeah, please.
That's a very large philosophical question that really should be done over a glass of wine, I'm sure. So I think the challenges in antimicrobial research at the moment are there are many challenges. I think there are lots of some of the big challenges being that big pharma has moved out of this area, and that's, I think, really more to do with the cost model. And there are lots of biotechs and smaller companies that are trying to bring assets forward, which then has its challenges in terms of understanding the full drug development process and how to get a really good target or idea or chemistry through to an actual clinical candidate can be hard to negotiate. There is, antibacterials have issues in the sense for toxicity, because often they require high exposures, and therefore you do require you are close to that what's tolerated for that toxicity profile. But I don't think it's the tox alone that's the issue. I think it's the cost model. I think it's the clinical route to get to the really the patients that really need the disease is not well established. So I think it's tricky. But I think with a lot of the work that's going on currently with CARB-X, etc., there's a lot of emphasis to try and get new bacterials forward, and I think that's a really positive thing. And I think we've learned a lot from the previous years of working in these areas as well. So I think maybe we understand some of the pitfalls better as well, to help get new things forward.
So thank you very much for taking the time to answer our questions. And I know some are not that easy to answer, and we all would like to get some recipes or cookbooks, but the field is really challenging and complex, and has to be looked at very individually, and they don't behave like we would have them. We would prefer them to behave. So with this last question and response by our toxicologist expert Claire Sadler, I'm closing this question and answer part of the webinar, and I'm handing over to Dagi and Astrid for some final comments. Thank you very much.
Thank you, Ursula, for moderating this session. And of course, thank you, Claire, for your time and for sharing your expertise about this, I see, very challenging topic. And also thank you to everybody on the line for joining the webinar and for actively contributing to the discussion with all your questions. Make sure to join us again for our upcoming webinars. We aim to host a webinar with Heinz Moser on the importance of physicochemical properties of antibiotics. Hopefully in December, we will keep you updated on the Revive website. And in January, we will have a webinar with David Schleis, Ian Friedland, Suma Tinambir, and Mayor Powell, this time about the development of antibacterial drug enhancer combinations and non-traditional products. You can already find the registration links for the webinars on the Revive website. I hope you found this webinar useful, and I would appreciate to receive your feedback in the survey, which will pop up after closing the webinar or by email. I hope to see many of you again for our future webinars. Goodbye.